So today we're going to be talking about how the cell makes a protein from the genetic code of DNA. This occurs in the nucleus and at the ribosome. So it's a two-part process. So how do we get from DNA, which codes for everything, to a protein, or what we look like? All right. First, the cell has to read the DNA code, and it has to transcribe it into a different format, which is going to be RNA, mRNA, and then it's going to be translated into a protein. So we've already learned DNA. We know that DNA is double-stranded. We know the sugar is deoxyribose, and there's 4 bases, A, T, G, and C. A and G are the purines, the bigger bases, and T and C are the pyrimidines, which are the smaller bases. There's also RNA. RNA is single-stranded, still made up of nucleotides. However, the sugar is ribose, and we just have a slight difference right here in what the sugar will look like. And the bases are A, G, C, and U. So the difference is U. There is no thymine in RNA. So instead, uracil is there. So uracil is a pyrimidine. Okay, so the process of transcription. This is the process in which we're making mRNA. This is going to occur in the nucleus, okay, of a eukaryotic cell, and then the cytoplasm of the prokaryotic cells. It's regulated by operons, which is in bacterial cells, and we're going to learn about that in another PowerPoint, or in eukaryotic cells, it's by transcription factors. And it does involve 3 different steps: initiation, elongation, and termination. Now, this whole process was cracked or discovered in 1961 by a guy named Marshall Nirenberg. All right, he made mRNA by adding the necessary components, and he found out that it coded for an amino acid called phenylalanine. Okay, this is called the central dogma theory. It's the flow of information from DNA to RNA to protein to trait. So that's kind of what we're learning. So the first part of transcription is initiation. Initiation is a start. Then elongation makes an mRNA molecule, and then termination ends it. So in initiation, RNA polymerase is going to attach to a promoter region on the DNA strand. Now, helicase, just like we saw that with DNA replicate, it's going to break the bonds and unzip the section of the DNA. And it typically will expose about 10 bases at a time. Free RNA nucleotides are going to move in, and they're in the nucleus, and they're going to bond to the DNA strand. Okay? Now, the promoter region, this region right here, okay, this is a short section of DNA that marks the start of a gene. All right, every gene on a chromosome has its own promoter. Okay, and that kind of defines the start of transcription, the direction of it, and the strand that's going to be transcribed. So for a eukaryotic cell, the promoter region is called the TATA box, T-A-T-A, which is about 25 nucleotides up site from an initiator site. Okay, now, either strand of the DNA template can be transcribed. So it's all based on this promoter region. Elongation is the process of making the mRNA. And again, it's made in the 5' 3' direction. Okay, so it's just base pairing of RNA to the DNA. And remember, in RNA, there is no thymine. There's a U. Now, when the RNA polymerase actually finds the terminator region or the stop region, it will then detach the mRNA, and it will have that released, and the DNA will go back together. So the terminator is a short section of the DNA that marks the end of a gene. And in eukaryotic cells, it's AAT, AAA. Okay? Now, an mRNA molecule may contain as many as 200,000 nucleotides. On an average, a human will have about 5,000 nucleotides. Okay? So, several copies of RNA are typically made at a time. All right, so if this is the DNA template, you want to figure out what the RNA is. And remember, when you base pair, there is no thymine when we're making mRNA. So that is the answer. All right, and all the RNA molecules that are going to be made are going to be identical because it's made from the code of the DNA. Okay, so mRNA is what is made in transcription. This is messenger RNA. Okay, this is going to take the message from the DNA and it's going to take it to the cytoplasm, take it to the ribosome in order to get translated into a protein. Every 3 bases is going to be a codon. All right, so these 3 bases are a codon, and that's what's going to code for a particular amino acid. Now, our RNA also exists. And hold on one second. Our RNA is called ribosomal RNA, and that's going to hold the ribosome together. If you see that the ribosome is made up of a large subunit and a small subunit. And this is very important because the mRNA is going to go in the middle, and the rRNA needs to be holding this together. A tRNA is a transfer RNA. It's highly wound together, and we're going to be dealing with this structure when we talk about translation. Now, I want to point out that we have a site that's called an anticodon, which are going to be 3 bases that are complementary to the mRNA. And then we have an area that's going to hold an amino acid, which is the whole point of translation, is bringing in the amino acids to the ribosome. Okay? Now, once we make the mRNA, we have to process it. It's a little long. So the process of shortening it, okay, is to add a methylguanine cap to one end and a poly A tail. Now, a methylguanine cap, think about it like out in the wintertime. You need to protect your head from the cold or the weather, right? So this is the same thing. We're kind of protecting the mRNA to get to the ribosome. Now, the poly A tail, think of it as a way to get there. And that's just 100 to 200 different adenines that are added on. Now, both of those signal in a signal to the cell that, hey, we need to shorten this mRNA. So the introns are removed, okay? The introns are non-coding portions of a eukaryotic gene. Those are the things that get removed. The exons are expressed, or they stay, and they get spliced back together. And if you can see here that the introns are removed and then the exons are joined together. Okay? In lower eukaryotic cells, so not very complex, they get removed by themselves. They have like a self-splicing mechanism. But in higher eukaryotes like us, we have what's called a spliceosome or a ribozyme that's going to help in doing that, because it's just a little bit more complex. Okay. Now, it takes only about 1,200 nucleotides to code for an average size proteins of about 400 amino acids. So we have a very long mRNA that's made, and we do need to shorten it so we can code for the particular protein. So that is transcription, the process of making mRNA. Once we've made it, we need to process it and shorten it. Now we can go through translation, which is the process of taking the amino acids that code for the mRNA which was made. And again, amino acids joined together make up a protein. All right, so we need the mRNA that was made, we need tRNAs which bring in the amino acids, and we need the ribosome and obviously our RNA that holds it together. And again, this is broken down into initiation, elongation, and termination. Initiation starts the process. Elongation brings in amino acids and elongates the polypeptide, and termination stops it. So initiation, all right, we need to have a start codon on the mRNA. So it will search along the mRNA at the ribosome until it finds the start codon. So this is what's happening. We're scanning, we're scanning, we're scanning, and then all of a sudden, right here, we see the AUG. And now we can go into elongation. Now a tRNA will bring in an amino acid which for AUG codes for methionine. And again, this is the tRNA molecule. This right here is the anticodon, and this is the amino acid. And it does look for the start codon. Now, the elongation is going to bring in the tRNAs to bring in the amino acids. Okay? So if you see what happens here is we had our first tRNA bring in an amino acid. This is called an A site because it has just one amino acid. And then we bring in another amino acid. So the anticodon is going to bond temporarily. It's going to bring in another amino acid. Now a bond is going to form between these two guys. And then this tRNA is going to leave. So this is then going to turn into the P site. Okay? And then that's going to allow it to move down the amino acid, the mRNA, one codon at a time. So if you see this one leaves, then we join this together, another one comes in, all right, this one will leave. So as one comes in, one leaves. And again, the peptide bonds are going to form. This is called the P site because it's holding a growing polypeptide chain. So tRNAs are going to be added and lengthening the polypeptide. And this occurs until we hit the stop codon, which is when we hit termination. Now there's three different stop codons, UGA, UAG, or UAA. Okay? Now as far as this goes, once it hits that stop codon, it is going to release the tRNAs, and the polypeptide is then going to be there, and it will lose that. It will then form the protein folding and so forth. It will use that. Okay. This also requires a protein called a release factor, which binds to the stop codon and also cleaves that polypeptide from the last tRNA molecule. Now, several copies of the polypeptide/protein are going to be made at a time. And they can be altered before they can function. Remember that proteins need to keep being processed, okay, before they can be ready to be used. Okay, so you're going to be using the chart in order to figure out the translation of this. So again, you need to figure out where the start codon is. So you scan along until you find the codon AUG. And we're going to have our little tRNA come in. The anticodon is going to be UAC, and this, based on the chart, is going to bring in the methionine. Okay? And then another tRNA is going to come in. It's going to have another anticodon, and this is going to bring in, based on the chart, it's going to bring in alanine. Okay, and this is going to happen. Remember, a peptide bond forms, this gets removed, and then we kind of go along and do that. So when we do this, I'm sorry that I wrote over it, but this is the process of forming this particular amino acid. Now, once it gets to UGA, that's our stop codon, and that doesn't count for anything. Okay, so the central dogma theory is going from DNA to RNA to protein to gene, and that's what protein synthesis is. Okay, so we take the DNA, we transcribe it into mRNA. Every 3 bases is a codon, and then that gets translated and turned into amino acids or the proteins. So you do need to learn how to use the chart, and that'll be another PowerPoint for you to see that. If you have any questions, please let me know.